Hi, this is KKR sir, Biology NEET faculty. Today we are explaining about the vectors in recombinant DNA technology. Vector, what's vector? Vector is a DNA molecule. It carries the foreign DNA into the host organism. It's a vector. Vector is known as vehicle DNA. Yes, it's correct. Here accession region. What is accession region? देखो accession vector is a vehicle DNA. Reason vector is carry the foreign DNA into the host organism. Four options. Accession region correct 2. Reason is the correct explanation for accession. Statement 1. Accession region wrong. Accession region both are wrong. Statement 2. Third option. Accession correct 2, reason correct 2. Reason is a not correct explanation for accession. Third option. Accession correct 2, reason wrong. Fourth option. What is the answer? First answer. Chances to come. Accession, reason correct 2. Reason is the correct explanation for accession. Then cloning vector. What is cloning vector? Cloning vector, the vector which is used for cloning of desirable genes, is called cloning vector. There are two types of vector. Natural vector, artificial vector. Natural vector, plasmids and bacteriophages. Artificial vectors. What is artificial vectors? Artificial vectors are PBR322, PUC109, BAC, bacterial artificial chromosome, YAC, yeast artificial chromosome, TA plasmid, tumor-induced plasmid, cosmids, phasmids. These are all our artificial vectors. Okay. Now, natural vectors, some important points. What are plasmid? Plasmid, extra chromosomal circular DNA present in bacteria is called plasmid. It having replication capacity. Next, bacteriophages. The virus which infect the bacteria. Point to be important. The bacteria which is not infect the virus. The virus which infect the bacteria is called bacteriophages. It controls the high copy numbers of their genomes. It controls the high copy number of cells. Bacteriophages. If less number of copies, desirable genes, multiplication required, we go with the plasmid. A well-known vector is plasmid. Okay. Now come to salient features. What are the salient features of vector? There are three important salient features are there. One is ORI. Second one, selectable marker. Third one, cloning sites. First one, ORI, origin of replication. Origin of replication. Where replication need to start? Controlled by ORI. How many copy number of their genomes or how many copy number of their desirable genes? Controlled by ORI site. Selectable marker. What is selectable marker? Selectable marker used to eliminate, identify and eliminate non-transformants and allow the growth, permit for transformants. Selectable markers, antibiotic resistance capacity they had. For example, ampicillin resistance, kanamycin resistance, tetracycline resistant, chloramphenicol resistance. These are all selectable markers. Cloning sites, third one. Very few, very important. Very few cloning sites required for multiplication of desirable gene. Generally, very few or only one cloning site is preferable. One cloning site have two antibiotic resistant genes. I repeat, one cloning site has two antibiotic resistant genes. Of course, more cloning sites there, it's complicated for the cloning. I'll give the one example. When you take the PBR322, उसमें BAM-H1 is the cloning site having two antibiotic resistant gene, tetracycline resistance and ampicillin resistant genes. These are the general features of cloning site, cloning vector. Thank you.